range.
Sir, share content.
तिमीले आफै शेयर गर्न मिल्छ होला नि त?
मिल्दो रहेछ। सर भयो है?
आयो ठिक छ।
ऋषभ सुरु गरौँ?
सो म स्टार्ट गर्छु है त।
ऋषभ तिम्रो आवाज एकदम अलि साउन्ड आइरहेछ है।
अहिले चाहिँ साउन्ड आइरहेछ?
अहिले त साउन्ड चाहिँ मिलाऊ है ऋषभ, तिमी ठ्याक्क अहिले चाहिँ आइरहेछ साउन्ड।
आई थिङ्क अब आउँछ होला है। Okay. So good morning, everyone. I would like to welcome in the presentation of gene therapy. So which is presented by Isa Grung and me myself. Okay, so let's begin our presentation. So gene therapy is basically a very emerging technology, and then it's very promising future as well. And talking about gene therapy, I personally love it and because I do have lots of interest in gene therapy. So let's begin with next slide, please. So okay, let's begin with a quote by George Mendel. I'm convinced that it will not be long before the world will acknowledge the result of my work. So he was the pioneer of genetical engineering, and he has contributed a lot in genetic engineering. So gene therapy is basically, next slide, please. All right, so here is the timeline of the gene therapy. So gene therapy is very promising future, and we are, I would like to, I would glad to say that we are in the golden era of the gene therapy. So now here is the, like in the slide, you can see that I wish I could get rid of the diseases, and in the 2050, in the upcoming days, in the 2050, there will be less expensive gene therapy, less expensive and effective gene therapy. So next slide, please. Would gene therapy cure all the illness in the future? So yes, this is a very crucial question, that the gene therapy will cure all the illness in the future, because gene therapy is being a very promising future as well. So maybe next slide, please. Okay, now finally we came to the introduction. So what is the gene therapy? So talking about the gene therapy, it is when the nucleic acid, when we insert nucleic acids into the cell and altering its property, and we cure the medical conditions or disease, the lack of comfort in our body. If we feel that gap, or if we feel that defective genes, so we gene therapy really works to fill that gap, and to provide the solution of the diseases. So it is the correction of genetic defects by transferring the normal functional gene copy of gene into cells. So what we basically do in the gene therapy is we just edit it or recombine the gene of the correct copy, functional copy, and we fill that gap, which gap has been created due to several mutations or disease conditions, or maybe something like that. So yeah, so the gene therapy, first gene therapy was the drugs was like approved in 1990. 1990 for the disease ADA, SCID, which is adenosine deaminase, immune deficiency. It is basically immune deficiency, severe combined immune deficiency disease, in which the patients have several immunity issues. Next slide, please. All right. So gene therapy, basically in our body there is two types of cell. So first is the somatic cell. In the somatic cell, we have the general growth of the cell, and there is a meiotic cell division of the in the somatic cell. And there is second is the germline cell. So what basically the germline cell is? So germline cell is, the term itself calls a germ. Germ means germination. So where the creation of the life or germination begins. So in the gene therapy, basically we do have two types of gene therapy: in vivo and ex vivo gene therapy. To explain furthermore, I would like to hand over this virtual mic to Isha.
Thank you, Rishabh. So now let's go over the types of the somatic gene therapy, which are basically the in vivo gene therapy and the ex vivo gene therapy. So for in vivo gene therapy, what happens is that we take the therapeutic gene, which is the gene of interest, and then we incorporate with it the viral vector, and then we introduce it into the patient's body by cell-specific direct injection into the tissue in need, where the transduction takes place inside the target cells, and the target cells encodes the production of the needed protein. So for the ex vivo gene therapy, what happens is that we take the stem cells, we isolate the stem cells from the patient's body, and we culture it in the lab, which is the via in vitro process. Then we take the therapeutic gene, which is the gene of interest, along with the viral vector, and then we also put it in the culture media. So the transduction takes place in the lab in vitro, and then we introduce the transduced cells inside the patient's body. So basically, in vivo, there are no specific target cells. I mean, specific target tissues as in ex vivo. For ex vivo, it only works on the bone marrow and the hematopoietic stem cells, whereas for in vivo, it's available for most of the cells at least. Blood cells, brains, lungs, skin, silver. Uh, so in vivo is used in the eye diseases like the liver congenital amaurosis, the neurological disorders, as Ishav said in the previous slide, ADA, SCID, spinal muscular atrophy, and hemophilia. So for ex vivo, it is a little narrow. It is a little narrower than the in vivos. It's used in immunological disorders and cancer types. So for ex vivo, it has a high transduction efficiency, and it is also less immunogenic than in vivo because the transduction takes place outside the patient's body and not inside like in vivo. So let's go over to the next slide. So here we have the methods of gene delivery. So here we have the methods of gene delivery. Firstly, we will go over the viral vectors used in use today for gene therapies. So here we have the retrovirus. Retrovirus was actually the first ever viral vector used in the first ever gene therapy clinical trials. We all know what retroviruses are. It uses RNA as its genetic material. It affects the cancer. It has this basically it affects it does cancer and tumor growth, HIV in patient's body or in the host's body. So it has oncogenic properties. So it was actually extensively used in the previous gene therapy trials, which was in 1950s to 1990s and all that. But then what happened in 2002 was the fact that a human trial for SCID, the bubble boy disease, was conducted, and 9 infants with SCID were cured via retrovirus-mediated gene therapy, which was an unequivocal success of gene therapy. But unfortunately, 4 developed leukemia, and one of them died, which was the reason was the oncogene property of retrovirus. So retroviral vectors basically has this U3 enhancer, which induces leukemia by activating expression of oncogenes near the chromosome insertion site. So the trials were stopped because retrovirus was not, then after retrovirus was not the preferred tool for the gene therapy. Then we have lentivirus. So lentivirus is also the genus of retroviruses, which is basically the cause for HIV/AIDS and all that, and it infects both dividing and non-dividing cells, whereas retrovirus just affects dividing cells. So as for lentivirus, it has long-term expression of the transgene. It has larger capacity, 9,000 BP per 9,000 BP per article. And then we have AV, the adeno-associated virus, the revolution of the gene therapy, the revolutionary gene tool used for gene therapy nowadays. So AV virus is the much preferred and the most safer tool compared to the two before, because it has no viral genes, it has low immune response, it has long-term gene expression. But then let's go over to the next slide. I'll explain about AAV more in the next slide. So we have the non-viral based vectors. I don't think I need to talk much about this because Sunil sir explained about this pretty much in our classes. So we have the plasmid one, then we have liposomes, and then we have nanoparticles. So why do we prefer viral vectors more than non-viral vectors, even though non-viral vectors do not induce cytotoxicity and immunogenicity and are much more biosafer than the viral vectors? Well, the fact is that non-viral based vectors are poor method of delivery of the needed gene. It has low expression of the transgene, and it was the clinical trials between 2004 and 2013, where non-viral based vector-based gene therapy tools were extensively researched, but then we again come back to the viral vectors because it is the preferred tool, because it transduces cells much quicker, and it is more efficient in delivering the genes in the targeted cells. Why do we use viral vectors? Yeah. As I said, it is the obligatory intracellular parasites, can only replicate inside cells, efficiently transfers viral genome into host cells where it is replicated, infects specific types of target cells. So these are the reasons why we use viral vectors. So virus AAV as a gene therapy vector. The fact is that we used to use AV, the adenovirus, the recombinant adenovirus. It was extensively used before, but then the results from animal models, it was not satisfactory, and there was a strong CD8 plus cytotoxic T cell response to the RAD gene expressed in transduced cells. So then after the AAV adeno-associated virus came in use, and then it's still the most preferred gene delivery tool now. So why do we use AAV as a gene therapy vector? So it does not cause disease. As I said, it's very poorly immunogenic. It infects both dividing and non-dividing cells. And AAV DNA genome generally persists in an extra chromosomal state, does not integrate into the DNA genome of the host cell. So when you insert the AAV DNA genome, it does not integrate into the DNA genome of the host cell. So how it works is that this is the structure of the AAV vector or the AAV virus. So here we have the ITR, and here we have the REP and the cap. So so the ITR helps in the replication of the this virus. This REP is also responsible for the replication, and this cap is responsible for encoding the capsid of the virus. So basically, it protects functions as the viral envelope of the virus. So when using this AAV vector, when inserting it into the host cells or the target cells, what happens is AAV vectors is packaged into three different packaging materials. So basically, we have how we use AAV as a recombinant or as the AAV viral vector is we remove this REP and CAP, and then we insert the gene of interest in this, but then basically making it replication deficient, meaning it does not replicate inside the target cells. But then we still need the REP and CAP, because CAP acts as the viral envelope, and the virus cannot enter the host cells without the cap. And we still, so what happens is we package this REP and CAP as the helper plasmid, and it is provided in trans. So for the third packaging, we have the adenoviral genes for AAV production. So basically, we have three packaging plasmids. We have the packaging plasmids for the gene of interest. We have the packaging plasmid for the REP and CAP, the helper provided in trans, and we have the packaging plasmid for the adenoviral genes. So let's go over now. Rishabh, I hand over the virtual mic to you.
Thank you so much. Okay, so time and again now. Okay, so what are the diseases approved for the gene therapy? So there are several diseases. FDA has approved the drugs for the several diseases. So first diseases is liver congenital amaurosis. So this disease basically is a diseases of blindness, and there is a mutation in 22 gene number, and it is the retinal degenerative disorders. So I will furthermore explain this in detail as well. So and then the second disease is ADA SCID, which is basically the uh, immuno, immunogenic deficiency disease, in which the patients doesn't produce a sufficient amount of B cells and T cells. Similarly, spinal muscular atrophy. So in this spinal muscular atrophy is there is problem in the muscular like contraction and relaxations. It basically happens in the infant babies, and likewise there is Wiskott Aldrich syndrome. So in this disease what happens is basically in the leg area there is swelling, and there is a pain and swelling in the leg areas and the hand areas. You can see the ashes and all the painful things over here. So let's move forward to next slide. Okay, so liver congenital amaurosis, LCA, what is this disease? It is an inherited retinal degenerative disease characterized by several loss of vision at birth, associated with eye-related abnormalities, including roving eyes movement, deep set of eyes, sensitive to the bright light, and visual acuity, usually limited to the level of counting the fingers and detecting hands motions and bright lights. Most most are autosomal recessive mutation. Mutation in 22 genes can cause LCA, accounting for 75% of LCA. Now, estimated birth prevalence of LCA is 2 or 3 per 100,000 birth. The common diseases caused of inherited blindness in the child. So, what is the cure of these diseases? So, next slide, please. Okay. So, Spark therapeutics treatment is for LCA and LCA2 caused by mutation in the RPE65 gene and affecting the retinal pigment epithelium. So basically it is caused due to the mutation in 22 number genes. Uh, Nonetheless, a gene therapy is designed to replace one mutant genes RP E65 in the principle be easily adopted to replace the different genes. So it's basically becomes a very magical and like um, miraculous in the terms of science and technology. So basically effects in metabolism of the visual pigment retinal, a form of vitamin A in the photoreceptor in the retina, the rod cells and cone cells, as well as in the immediately adjacent these the retinal pigment in the epithelium. It affects in the mainly retinal pigment of epithelium, so that the patients could not the patients became sensitive to lights, and they couldn't count their fingers, and all the seeing stops, viewing the views stops. So basically, so how we do this, like the method of how we do this. So we access the retinal and visual function with the RPE65 associated liver congenital amaurosis, given one subretinal injection of adeno associated virus containing a gene encoding a protein needed for the uh, isomerohydrolyze activities of the retinal pigment epithelium. So we basically give this in the form of certain doses, and due to that certain doses, the patient uh, maybe in the 6 month the patient gain their sights, um, like sights in simultaneous way, or maybe slowly they are getting their sights. So next slide please. All right, here you can see the drugs approved by FDA. So there are the list of drugs in vivo gene therapy and ex vivo gene therapy. So basically, there is Glybera and Lutexnia. So this Lutexnia, what it is, it does in the previous disease we talk about the liver congenital amaurosis. This disease, gene therapy used in that disease as well, and we could see the lots of gene therapy drugs in vivo and ex vivo as well. And I hope that you will go through it when we will provide the slides about that. So next slide, please. All right. So I would like to give this virtual mic to Isa again. Thank you.
Okay, thank you, Rishabh. So now let's go over the ongoing clinical trials for hemophilia. So those the diseases Rishabh talked about, it was actually the pretty much a success for the gene therapy. So now we are talking about the ongoing clinical trials for gene therapy, which is the hemophilia. So we all know what hemophilia. It is the inability to form blood clots. So it is of two types, hemophilia A and hemophilia B. So it's also known as the royal blood disease. So basically, the daughters are the carriers, and it's also a result of the inbred in the the royal family and all that. So yeah, the hemophilia A is caused by the defect in factor 8, and the hemophilia B is caused by the defect in factor 9. So F8 and F9, factor 8 and factor 9 are both expressed by the liver. And so for this factor 8 and factor 9, I think we need to go a little back to the third semester where Samiksha ma'am taught us the coagulation cascade, the intrinsic pathway and the extrinsic pathway, and the which factor activates which factor, leading to the expression of the thrombin and fibrin, basically the enzymes used in this clotting factor. So factor 8 and factor 9 is basically the defect genes that induces hemophilia. And the tool of the gene therapy tool that uses hemophilia is the AAV. As I said in the previous slides, AAV is the most preferred tool, and hemophilia utilizes different modified forms of AAV. So hemophilia is an X-linked disease, meaning the daughters are carriers, meaning it is dominantly persistent in males, is seen in males. So let's go over the hemophilia B clinical gene therapy trials. But before that, I would like to give you a little backstory on how hemophilia was previously treated. So for hemophilia, what used to happen was the initial treatments of hemophilia, there was transfusion of plasma. Okay. So it was very inefficient because large volumes were needed to achieve a level of the factors being replaced. So then after the more efficient therapy, which was the factor replacement therapy, came. So for this, the clotting factor concentrates were injected. So basically, the clotting factor concentrates, if you were infected with the hemophilia A, if you had hemophilia A, you infected clotting factor 8, and if you had hemophilia B, you injected clotting factor 9. And it was a very prevalent during the 1980s and the 1990s, where the HIV epidemics and pandemics were going on, the AIDS, the hepatitis. So it was this factor replacement therapy. So what basically it was a home treatments, because you needed to inject it 2 to 3 times a week, because the factors, it had very short half lives. It had very short half lives, and the factor for factor 8, it had the half life of 10 to 14 hours, and for the factor 9, it had the half life of 15 to 24 hours. So you needed to inject it throughout the week, or throughout your life. It was very expensive. So since you basically injected you needed a donor, and you injected it into your blood, or something like that. And then what happened was, during 1980s, the AIDS and the hepatitis, it was very prevalent, so it hampered the fact that injecting the clotting factor into your body made the chances of you being infected with hepatitis C virus was 90%, and you being infected with HIV was 55%. So you are gearing towards getting your hemophilia under control, but then you also get the side treats, the side dishes, which were the AIDS, hepatitis, and all that. So, in 1990, genetically engineered recombinant factor therapy became prevalent, which drastically reduced the risk of the factor therapy, because before that there was no checking of the blood transfusion and all that. So after doing all that, so it drastically reduced the chances of you being infected with HIV/AIDS and all that came along with injecting clotting factor inside you. But then still, it did not take away other side effects, which were the inhibitor antibodies. Because then after hemophilia, it's also treated by the supplementary, treated the supplementary missing clotting factor by injecting it inside your body. But then but then the fact was that the body, your body, it produced the inhibitor antibodies against the clotting factor. So for hemophilia B, it was 1 to 6%. Your body produced antibodies against it. But for hemophilia A, it was 20 to 32%. So there were very severe bleeding episodes, intracranial hemorrhage, joint diseases, and all that. So what happened? Gene therapy is the ultimatum for all of these side effects of treatment of hemophilia, because firstly, the replacement therapy treatment is very expensive, and you have to inject it for a lifetime. And also the antibody respond to clotting factors, so it's not a win-win situation. So the gene therapy can eliminate the challenges associated with factor replacement therapy. It restores the functional copy of the affected gene, which could potentially ameliorate the clinical manifestation of the disease. So here we have the hemophilia B clinical gene therapy trials. I'm not focusing on hemophilia A, because even I could not understand the depth of it, since is that the hemophilia B is caused by the defect in the single clotting factor, the single gene, that is factor 9. But then for hemophilia A, what happens is the von Willebrand factor, which basically binds and carries factor 8 along to the circulate, and it circulates in the bloodstream. So there you have the two genes responsible for hemophilia A, but for hemophilia B, there's only one gene. So there's a defect in a single gene. So let's focus here. These are the hemophilia B clinical gene therapy ongoing trials, and which are this AMT061, and this is the FIX FORA AAV5. This is the modified version of the adeno-associated virus, and it is in the clinical trial stage 3. And the sponsor is the Unicure CSL Burning. So as you can see, number 2, number 3, number 4, number 5, number 6. If you are interested in it, you can go over it when we provide the slides to you. So let's go over the AMT061, which is the most, which has the most, the success rate, which is more likely to be successful. So AMT061, why is this slide on here? So AMT061 is an experimental gene therapy that Unicure is developing to treat hemophilia B. So AMT061 uses a modified and harmless adeno-associated virus, AAV. Excuse me, can you see the words I have written in this to deliver here? Or is this blocking you too? Hello, Rishabh? Bye. AMT061 uses a modified and harmless adeno-associated virus, AAV5, virus 5, AAV5, to deliver a highly functional copy of the F9 gene called the FIX SPORA to patient cells. So the fixed SPORA gene version was shown to result in FIX clotting activity 8 times greater than the associated with the standard F9 gene. As such, the one-time therapy administered directly into the bloodstream is expected to increase FIX level, helping to prevent and control bleeds. So we just don't basically call gene therapy our future, because if this trial succeeds, if it is succeeded, then the fact that you don't have to inject the clotting factors all over the life, and all the money that goes into it, and all the this associated with it, it goes away. So yeah, this is the if this successes, then if it succeeds, then it is the revolution. Yeah. Also, the Unicure, the 2017 was considered the important year of gene therapy, and is also considered as the launch part for the new era of modern gene therapy. So this AMT061, which is basically by the Unicure company, is actually guessed to be succeeded at, I think, 2025 or something like that. But you can go over it if you want to. So how does this works? So as you can see, it is the AAV vector here and the AAV vector uptake. So here is the when the AAV vector is uptaken by this skin. So what happens is it forms the endosome. It forms the endosome, and there is a nuclear entry. And when it is about to enter the targeted cells, then the capsid is uncoated. The capsid is, let's say it goes, and then here the synthesis of the transgene of the second strand. So here, once the capsid uncoats, then the synthesis of the transgene happens, and then the transgene is expressed, and the transgenic protein synthesis happens, meaning the required proteins that you needed by the gene is secreted, and the secretion happens. So these are the key companies, the key players in the gene therapy. So here we have the Unicure company, the Resens Bio Inc., the Orantis Therapeutics Inc., Solid Biosciences Inc., Mira GTX Holdings PLC, and all that. These are the key players in the gene therapy, and the market is expected to reach $12.16 billion in 2025 at a CAGR of 32%. So yeah, gene therapy has a huge potential. And if you guys want the money as well as the the drive to save humanity or something like that, then you can also you can also do your major in gene therapy.
Thank you so much. I would especially would like to thank Isa for giving a special effort in making this presentation. And thank you so much everyone for listening patiently. Over to you, sir.
Okay, thank you very much. Uh, तिमीहरूले चाहिँ एकदम मिहिनेत गरेर तिमीहरूले बनाएको रहेछौ होइन that makes me happy right you really put your effort into it अनि तिमीहरूलाई चाहिँ मलाई के तिमीहरूलाई अलिअलि टिप्सहरू चाहिँ मैले तिमीहरूलाई भन्नुपर्ने हुन्छ यहाँ so that's good that is good होइन तर इन्फर्मेसन वाज अल्सो गुड तिमीहरूले चाहिँ भनेको जुन एनर्जीले तिमीहरूले भन्यो द इन्फर्मेसन यु ब्रट हियर वाज भेरी गुड है तिमीहरूको स्लाइडमा मैले चाहिँ जस्तै नि अब राइट फ्रम द सुरुमा भन्दाखेरि चाहिँ रिसर्भले भन्दाखेरि होइन ग्रेगर मेन्डल भनेको जोन ग्रेगर मेन्डलको नाम लिँदाखेरि यु मस्ट टेक इट करेक्टली है भन्दाखेरि तिमीहरूले चाहिँ यो नेमहरूको प्रोनाउन्सिएसन राम्ररी लिनुपर्छ जोहान ग्रेगर मेन्डल हो क्या ऊ चाहिँ होइन रिसर्भले भन्दाखेरि जोन यसरी भन्नु हुँदैन रिसर्भ ठुल्ठुलो चाहिँ तिमीहरूको साइन्टिफिक कम्युनिटीहरूमा एटलिस्ट तिमीहरूले नेम चाहिँ करेक्टली लिनुपर्छ अनि ग्रेगर मेन्डल चाहिँ पायनियर अफ जेनेटिक इन्जिनियरिङ हो र ग्रेगर मेन्डल चाहिँ नि पायनियर अफ जेनेटिक इन्जिनियर हो र ग्रेगर मेन्डल है त्यसो यतिकै तिमीहरूले अब कस्तो भन्दाखेरि तिमीले अब चाहिँ इट सुड बी मोर स्पेसिफिक है तिमीहरूको चाहिँ तिमीले दिएको इन्फर्मेसनहरू कस्तो हुन्छ भन्दाखेरि म पनि यस्तै थिएँ म ब्याचलर्समा हामी पनि यस्तै थियौँ होइन यसो भन्नु पायो भने गफ हानिहाल्ने टाइपको बानी हुन्छ नि कसैसँग त्यस्तो चाहिँ हटाउनुपर्छ कि त्यसो मैले भने सुनिरहेको छौ
Yes, sir. I will definitely improve it.
तिम्रो चाहिँ नि यो यसै जस्तै मेरो विचारमा आई डोन्ट थिङ्क ग्रेगर मेन्डलले जेनेटिक इन्जिनियरिङ गरेर गयो जस्तो मलाई लाग्दैन है त्यो भेरी वर्ल्ड क्रसिङ गराए बट अब इट अभियसली इट ले द फाउन्डेसन है अभियसली इट ले द फाउन्डेसन फर जेनेटिक इन्जिनियरिङ बट वर्क डिएनएसँग हो तर चाहिँ नि तिम्रो आई डोन्ट थिङ्क अब ग्रेगर मेन्डल चाहिँ वाज अ पायनियर है पायनियर हो तिमीले भन्नु नि त अझ पायनियर है obviously he is a father of modern genetics होइन मेन्डल इन जेनेटिक्स इज अ फादर अफ जेनेटिक्स इज नोन एज अ फादर अफ जेनेटिक्स बट पायनियर अफ जी चाहिँ मलाई हो जस्तो लाग्दैन यस्तो स्टेटमेन्टहरू भन्नु भएन होइन अनि अनि भोकाबुलरीहरू युज गर्दाखेरि चाहिँ नि कस्तो भन्दाखेरि अब जस्तै अब जस्तै तिमीहरू चाहिँ नि कस्तो छ भन्दाखेरि ठिक छ तिमीले फ्युचर अफ जिन थेरापी इज भेरी गुड भनेर भन्यो द्याट्स गुड द्याट्स ओके होइन तर चाहिँ अब ईसाले पछि भन्यो एकदम ब्राइट फ्युचर भनेर एकदमै चाहिने एकदमै ड्याम हुन्छ नि 100% प्रमिसिङ फ्युचर जस्तो पनि छैन यसको अहिले टिल नाउ lot of clinical trials going on होइन lot of clinical trial goings on अनि तिमीले के भन्न सक्छौ इट क्यारिज अ बिग पोटेन्सियल भन्न सक्छौ है इट क्यारिज अ बिग पोटेन्सियल होइन फर न्यू थेरापिज होइन फर द ट्रिटमेन्ट इट क्यारिज बिग पोटेन्सियल फर द ट्रिटमेन्ट अफ न्यू डिजिजेस जेनेटिक डिसअर्डर्सहरू भन्न सक्छौ होइन तिम्रो चाहिँ अब गोल्ड स्ट्यान्डर्ड नै भनेर अनि चाहिँ तिमीहरूको गोल्ड स्ट्यान्डर्ड ट्रिटमेन्ट हो एक्चुली भएपछि तर चाहिँ स्टिल साइड इफेक्ट्सहरू लिमिटेसन्सहरू यसको धेरै छ एक्सपेन्सिभ छ यो होइन कि म्यासिभ क्लिनिकल ट्रायल हुनुपर्छ होइन त्यो पनि छ तर चाहिँ अब हामीले चाहिँ पोटेन्सियल चाहिँ ठुलो छ युज बिग पोटेन्सियल छ तर चाहिँ स्टिल हामीले चाहिँ जुन म्यानिपुलेट गर्ने जुन नलेज हामीसँग चाहिँ स्टिल द्याट इज लिमिटेड कि अझै साइड इफेक्ट्सहरूसँग चाहिँ के हो कसो हो सबै चाहिँ चेक गर्नुपर्ने हुन्छ होइन अनि तिमीहरूले भन्यो एडिएससिआइडीको इक्जाम्पल दियो ठिक छ अनि चाहिँ अब एचआइभीको पनि दियो ठिक छ होइन अब एएभीको पनि एक्सप्लेन इट होइन डज नट इन्टिग्रेट इन द जिनोम अब यो एएभी भनेको एडिनो एसोसिएटेड द्याट्स गुड द्याट्स नाइस है राम्रो लाग्यो एडिनो एसोसिएटेडको तिमीहरूले एक्सप्लेन गरेको है अनि तिमीहरूले चाहिँ के लेख्नुपर्छ भन्दा फिगर छ भने इफ यु आर प्रोभाइडिङ सम फिगर्स देन है फिगर यो फिगर 1 फिगर 2 यदि तिमीहरूको फिगर छ भने है फिगर 1 फिगर 2 भनेर फिगरको तलपट्टि लेख्ने है फिगरको केसमा चाहिँ तिमीले ध्यान दिएर सुन फिगरहरू छ भने चाहिँ सधैँ फिगरको चाहिँ तिमीहरूको नामाकरण फिगरको तलतिर गरिन्छ है फिगर छ भने अनि इफ यु हेभ टेबल्स अनि इफ यु हेभ टेबललाई चाहिँ माथिपट्टि लेखिन्छ टिएबिएलई टेबल 1 2 भनेर टेबलको माथिपट्टि चाहिँ लेखिन्छ टेबलको केसमा चाहिँ है फिगर चाहिँ सधैँ फिगरको तलतिर हुन्छ लेबलिङ है टेबल चाहिँ टेबलको लेबल गर्दाखेरि चाहिँ टेबलको माथिपट्टि गरिन्छ है टेबलको माथिपट्टि गरिन्छ कहिले कहिले विद्यार्थीहरूले चाहिँ यो सिम्पल कुरा हो सिम्पल तर चाहिँ दिस इज हाउ वी डु इट है हाउ डु इट तिमीहरूले चाहिँ यो ध्यान राख है अबदेखि सधैँभरि ध्यान राख टेबललाई फिगर ठिक छ तिमीहरूको चाहिँ त्यस्तो धेरै त थिएन बट स्टिल तिमीलाई मैले रिमाइन्ड गरेको है अनि त्यसपछि छ तिमीले चाहिँ त्यो युनिक इयरको पनि एक्सप्लेन गर्यो द्याट्स गुड अनि चाहिँ नि अब त्यो जिनवाला चाहिँ तिमीहरूको फ्याक्टर 8 र फ्याक्टर 9 स्ट्यान्डर्ड फ्याक्टर 8 र स्ट्यान्डर्ड फ्याक्टर 9 को कुरा गर्यौ अनि त्यो भन्दै गर्दाखेरि चाहिँ नि फेरि अब कस्तो पनि कहिले कहिले भन्नुपर्छ भन्दाखेरि तिमीले जिन थेरापीको यु ह्याड बेटर पुट द लिमिटेसन्स अफ जिन थेरापी है तिमीले जिन थेरापीको लिमिटेसन्स राख्यौ तिमीले जिन थेरापीको लिमिटेसन्स राखेनौ नि है
राखेर नि सर बिर्सेछ
है किनकि कस्तो होइन ठिक छ के फरक पर्यो नि तर तिमीहरूले चाहिँ नि किनकि यो चाहिँ स्टिल अन द ट्रायल्स है अनि सबै डिजिजको लागि स्टिल वी डोन्ट गो फर जिन थेरापी तर चाहिँ कस्तो छ भन्दाखेरि 100% क्योर हुन्छ भन्ने जस्तो चाहिँ स्टिल स्योर हुन सकिन्न कि कहिलेकाहीँ है त्यही भएर तिमीले यसको बारेमा राख्नु भए अझै राम्रो हुन्थ्यो लिमिटेसन्सको बारेमा कि है किनकि लिमिटेसन्समा राख्नुपर्ने कुरा छ कि यसमा है किनकि यसको अलिअलि तिमीले आफूले भन्नु त भन्यौ यस तिमीले भन्नु त भन्यौ होइन सधैँ सक्सेसफुल हुन्छ भन्ने छैन भने त भन्यौ बट टु बी मोर फ्यु स्पेसिफिक लिमिटेसन्सहरू राखेको भए अझै नाइस हुन्थ्यो अरू त ठिक छ त यु डिड वेल अनि तिमीले चाहिँ अलिअलि रिसभले चाहिँ अलिअलि उसमा वर्कआउट गर्नु पर्यो रिसभ आफ्नो भोकाबुलेरी र ग्रामरमा वर्कआउट गर्नु पर्यो रिसभ बुझेन भोकाबुलेरीमा पनि वर्कआउट गर्ने ग्रामरमा पनि है यो चाहिँ बेला पनि यही हो वर्कआउट गर्ने टाइम पनि यही नै हो होइन मोस्ट अफ द स्टुडेन्ट्स इम्प्रुभ ड्युरिङ देयर अन्डर ग्रेजुएटकै बेलामा तिनीहरूले इम्प्रुभ गर्ने हो सबै आफ्नो प्रेजेन्टेसन स्किल्सहरू भोकाबुलेरीहरू ग्रामरहरू म पनि ब्याचलर्स हुँदैखेरि अब अलिअलि सिकेको है फुल्ली त अझै पनि स्टिल वी आर लर्निङ स्टिल आई एम लर्निङ तर अलिअलि स्किल्सहरू साथीहरूबाट यताउताबाट आफूले वर्कआउट गरेर जान्ने सिक्ने भनेको चाहिँ अन्डर ग्रेजुएट दिस इज द टाइम यु विल लर्न है के छैन तर अलिअलि फोकस गऱ्यो भने झन् अझै सजिलो हुन्छ ए मैले यहाँ चाहिँ फोकस गर्छु है भन्यो भने इट विल बी इजियर होइन फोकस गऱ्यो भने आउँछ आफै त्यो अटोमेटिकली त्यही फोकस गरिन्छ मेरो स्किल्स मेरो भोकाबुलेरी मेरो ग्रामर मैले कसरी भन्नुपर्छ भन्ने कुरा थाहा हुन्छ अनि ओभरअल प्रेजेन्टेसन तिमीहरूको राम्रो छ तिमीहरूको एनर्जी द्याट वाज भेरी नाइस होइन तिम्रो जुन एनर्जी एकदम keep the same energy, keep the same enthusiasm, keep the same energy. Okay? That was very, very contaminating energy one. That's good. Energy त्यही राखेर अरू कुराहरू तिमीहरूले चाहिँ सच्याउ है. That will be fantastic.
Thank you.
यही नै भयो.
Thank you so much, sir.
Okay, you're always welcome. अब त्यसपछि चाहिँ नि अरू सुस्मिताको प्रेजेन्टेसन छ र सुस्मिता अरू कसैको प्रेजेन्टेसन बाँकी छ र मैले यसो भनेको
मेरो छ
ओके त्यसो भए सुस्मिताको पनि गरौँ न त आज प्रेजेन्टेसनै गर्ने भयो क्या ल है तिमीहरूलाई मैले चाहिँ क्लास अलिअलि तिमीहरू क्लास अलिअलि रस भयो भने तिमीलाई म एक्स्ट्रा क्लास लिन बोलाउन सक्छु तिमीहरू आऊ है अहिले त होइन अलि पछि के भयो त्यस्तै पर्यो भने होइन
साँच्ची मैले क्वेशन्स छ भनेर सोध्नै बिर्से सर एनी क्वेशन
सरी त्यही त अनि ठिक छ तिमीहरू कसैलाई केही क्वेसन्सहरू छ भने सोध बिफोर वी मुभ स्टार्ट द प्रेजेन्टेसन अफ सुस्मिता होइन इफ यु हेभ एनी क्वेसन्स तिमीहरूले सोध प्लिज द्याट्स भेरी इम्पोर्टेन्ट थ्याङ्क यू फर रिमाइन्डिङ तिमीहरूले क्वेसन सोध्नु इट्स अलवेज इम्पोर्टेन्ट है अलवेज अनेस्टली है तिमीहरू साथीहरू भनेर है कस्तो हुन्छ भन्दाखेरि ए म साथी हो म क्वेसन नसोध भन्ने होइन कि तिमीहरूले चाहिँ what you should change your mindset है तिमीहरूले चाहिँ प्रेजेन्टेसन बनाउँदाखेरि नै मेरो साथीहरूले मलाई कहाँनिर क्वेसन सोध्यो भने है मेरो मेरो पियर्सहरूले फेलो मेट्सहरूले मलाई कहाँनिर क्वेसन सोध्यो भने चाहिँ मैले कसरी यसको एन्सर दिने भन्ने प्रेजेन्टेसन प्रिपेयर गर्नेले पनि त्यसरी नै प्रिपेयर गर्नु पर्यो सोध्नेले पनि त्यसरी सोध्नु पर्यो है अब कस साथीले मलाई सोध्छ भने मैले के भन्ने होला होइन कसरी एन्सर दिने होला त्यो भ्यू पोइन्टले तिमीहरूले प्रेजेन्टेसनहरू बनाउनु पर्यो टाइम लिएर बनाउनु पर्यो र क्लासमा पनि सोध्नु पर्यो कि ताकि तिमीहरूको नलेज बाँडफाँड हुन्छ इन्टरेक्सन्स बढ्छ ब्रेन एक्टिभ हुन्छ है त्यही भएर तिमीहरू के क्वेसन छ यिनीहरूकोमा ऋषभ र के अरे ईसाकोमा प्रेजेन्टेसन के छ तिम्रो क्वेसन्सहरू के छ भने जस्तो आस्क है सोध त दुई तिनवटा क्वेसनहरू सोध त तिमीलाई के सोधेन बुझेन भने मलाई फेरि बुझाऊ भन मैले यो बुझिनँ भन जिन थेरापीले कसरी काम गर्यो होइन कसरी के भयो भन्ने सोध ल सोध एक दुईवटा सोध म सुन्छु तिमीलाई सोधेको सुन्छु म कोहीसँग छैन क्वेसन म तिमीहरूको क्वेसन पनि सोध्न म तिमीहरूलाई लगाउँछु है त ल मलाई चाहिँ कसले क्वेसन सोध्नु यहाँ क्वेसन सोध्नु पर्यो भन्दाखेरि रुबीले सोध्नु पर्यो रुबीले यिनीहरूलाई क्वेसन सोध्नु पर्यो ईशा र ऋषभलाई क्वेसन सोध्नु पर्यो रुबी एउटा सोध केही हेलो रुबी इज देयर
Yes, sir.
ल रुबी तिमी सबै बुझ्यौ भयो त ठिकै छ यसो के छ भने सोध बुझ के छ
छैन, sir.
छैन त्यसो भए रेजिनालाई केही छ कि
छैन, sir.
यतिकै ओहो कस्तो एउटा के सानो जस्तै भए पनि जिन थेरापीको बारेमा मलाई के चाहिँ इम्पोर्टेन्ट कुरा हो भन्ने भए पनि देवका
के अरे मेरो सर क्वेसन चाहिँ छैन बट एउटा चाहिँ यो जुन यिनीहरूले स्पाइनल मस्कुलर एट्रोफी भन्ने डिजिज भन्ने चाहिँ छ त्यो चाहिँ यो के अरे हाम्रो यो के अरे सोसियल मिडिया पनि एउटा सानो नानीको सियोना भन्ने नानुलाई एउटा यो डिजिज लागेर रहेछ होइन यो सायद सबैले थाहा छ होला अनि उसलाई चाहिँ अहिले सबैले हेल्प गरेर र चाहिँ एउटा के अरे एउटा मेडिसिन दिन लगाएको रहेछ क्या जोलजेजमा भन्ने अनि त्यो चाहिँ वर्ल्डकै मोस्ट एक्सपेन्सिभ ड्रग्सहरूभित्र पर्दो रहेछ कि अनि यसको कस्ट चाहिँ अराउन्ड $2.1 मिलियन रहेछ अनि ठ्याक्क मैले हिजो हेरेको थिएँ अनि आज त्यही कुरा भएर चाहिँ सेयर गर्न मन लागेको
ए ओके ल ल ल जस्तो त तिमीहरूले चाहिँ यसो हेल्प गर्न सकिन्छ भने अब तिमीहरूले गर्न सक्छौ भने डु इट आफ्नो ठाउँबाट होइन तिमीहरूलाई कि अब जस्तो त्यो अनलाइन क्याम्प पनि बनाउनु छ भने यु क्यान डु इट अब हेल्प गर्नुको लागि है फन्ड रेज गर्छौ भने तिमीहरूले गर्न सक्छौ बारेमा
फन्ड रेज भइसक्यो अहिले चाहिँ उसलाई चाहिँ ट्रिटमेन्टको उसमा लाँदै रहेछ कि अनि त्यसमा चाहिँ यहाँनिर रुटिन अफ नेपाल बन्दले चाहिँ सेयर गरेको रहेछ क्या अनि ठ्याक्क मलाई चाहिँ याद आइरहेको थियो अनि ठ्याक्क जिन थेरापीको टपिक आयो अनि मैले सेयर गरिदिएको
ए ल ल ओके अब स्पाइनल मस्कुलर एट्रोफीको बारेमा कसैलाई के भन्न छ त तिमीलाई के त्यो ऋषभलाई
ओके दिस इज भेरी
ऋषभलाई
एक्सप्लेन गर्नु पर्यो
एउटा के 1-2 लाइन्स भने पनि हुन्छ एसएमए को बारेमा ठिक छ अब ठिकै छ ल ल के छ भने भन ल
एसएमए पनि अटोसोमल रिसेसिभ हो अनि त्यही हो यसको सिम्टम्सहरू चाहिँ हाम्रो यो सिम्टम्स चाहिँ मसल विकनेसहरू अनि के अरे यो प्यारालाइसिस अनि इन्फान्टाइल डेथ हुने त्यस्तो हो कि अनि यसमा चाहिँ हाम्रो के अरे के हुन्छ भन्दाखेरि जुन स्पाइनल कोर्डमा चाहिँ अल्फा मोटर न्युरोन्सहरूको चाहिँ डेथ भएर भएको हुन्छ अनि एकदमै रेयर भनौँ न एकदमै रेयर हो यो अफेक्ट्स 1 in 6,000 लाइफ फर्स्ट भन्छ कि यसलाई चाहिँ अनि
प्लस यो चाहिँ के हुन्छ भन्दाखेरि यो चाहिँ
कुन जिनमा डिफेक्ट भएको त्यो चाहिँ
SMN1 gene, sir.
Autosomal recessive हुन्छ हैन
तिम्रो चाहिँ नि कतिवटा जिन्स रेस्पोन्सिबल छ यसको SMA तिमीले SMA1 SMA2 कतिवटा छ
त्यो चाहिँ थाहा भएन है सर यति थाहा छ कि
one SMA most severe type is one SMA leading to death before the second birth भनिरहेछ हैन so basically यो चाहिँ one SMA मा चाहिँ leading चाहिँ death गर्छ second birthday भन्दा पहिले नै मर्छ like 5 to 7 month भित्र चाहिँ मर्ने chances higher हुन्छ के बच्चाहरुको अनि इन्फ्यान्ट मा चाहिँ एकदम हाईली
अब चान्सेसहरु एकदमै यो देखा पर्ने बढ्ने हुन्छ नि त
अनि तिमीलाई थाहा छ अनि तिम्रो चाहिँ यो तिम्रो रिसेसिभ डिसअर्डर्सहरु भनेको के हो रिसेसिभ डिसअर्डर्स भनेको हाउ डु एक्सप्लेन इट अनि कुन चाहिँ डेन्जर हुन्छ तिम्रो चाहिँ
रेसेसिभ जिनमा इफेक्ट भएको होला
त्यो चाहिँ के कस्तो हुन्छ नि मेकानिजम हाउ यु हुन्छ तिमीहरूको चाहिँ अब रिसेसिभ जिन्स मा हैन तिमीहरूको चाहिँ नि कुनै डिजिजको केस चाहिँ डोमिनेन्ट मा हुन्छ अब अटोसोमल रिसेसिभ भन्छ अटोसोमल डोमिनेन्ट भन्छ अब यसो कम्पेयर गर्नु पर्यो भने अटोसोमल डोमिनेन्टहरू मोर डेन्जरस होला कि अटोसोमल रिसेसिभहरू मोर डेन्जरस होला
मेबी रेसेसिभ चाहिँ मोर डेन्जरस होला बिकज रेसेसिभ जिन चाहिँ इन्टरनली नै धेरै हुन्छ नि त्यसले चाहिँ मेबी धेरै नै वर्कआउट गर्छ बट आई एम नट प्रिटी स्योर अबाउट दिस आन्सर बट इट्स गेस भन्नु
इच्छालाई के भन्नु छ यसमा
सर रिसेसिभै होला किनभने हाम्रो हेमोफिलिया पनि रिसेसिभै हो अनि यसमा पनि अब त्यही एक्स म्युटेन्ट जिनले गर्दा हो आई थिंक रिसेसिभै होला
अलि भएन तिमीहरूले चाहिँ खोजिल तिमीहरूले यो आई गिभ यु असाइनमेन्ट है डोमिनेन्ट मोर डेन्जरस छ कि रिसेसिभ मोर डेन्जरस छ एन्ड वाइ एकदम सिम्पल 2 लाइनमा 3 लाइनमा एन्सर आउँछ तिमीहरूलाई मैले नेक्स्ट क्लासमा म तिमीहरूलाई सोध्छु यसरी हेरेर आउनु है कुन डिसअर्डर मोर डेन्जरस हुनसक्छ अटोसोमल रिसेसिभ फर्म होइन के अरे रिसेसिभ फर्मको जेनेटिक डिसअर्डर्स मोर डेन्जरस होला कि डोमिनेन्ट फर्मको रिसेसिभ डिसअर्डर्स मोर डेन्जरस होला प्लिज एभ्रीबडी तिमीहरू सबैले गरेर हेरेर आऊ है यो एक्चुली ओके अनि अरू केही क्वेसन छ अरूलाई एनी क्वेसन्स अरूलाई केही छ सुमित्रालाई केही छ
छैन सर
ओके त्यसो भए अरू कसैलाई केही छ सुमित्रालाई केही छ
मेरो दाँतमा इनामेल छैन त्यो जिन थेरापी गरेर फेरि आउँछ रम्बामा
तिम्रो दाँतमा इनामेल छैन अब जिन थेरापी गरेर आउँछ कि आउँदैन
अब योमा चाहिँ के हुन सक्छ भन्दाखेरि एउटा कुरो चाहिँ जस्तो हाम्रो एभी को डिफ्रेन्ट टाइप्सहरु छ है सो एभी ले के गर्छ भन्दाखेरि डिफ्रेन्ट ठाउँमा जस्तो लङ्सको लागि एभी8 छ हैन अरु डिफ्रेन्ट चाहिँ एभी को टाइप्सहरु छ हैन सो एभी ले के गर्छ भन्दाखेरि त्यो ठाउँमा चाहिँ त्यो रिसेप्टरमा अट्याक गर्छ हैन इफ सपोज तिम्रो चाहिँ लाइक इनामेल बनाउने प्रोटिनको हामीले त्यो एभी मा लोड गर्यौं अनि त्यसको भाइरुलेन्स प्रोपर्टी हटायौं भने अनि त्यसको क्लिनिकल ट्रायल इफ सेफ छ भने मेबी त्यो इन्जेक्ट गर्यो भने त्यो प्रोटिन यदि इनकोड गर्यो भने मेबी देयर आर चान्सेस कि त्यो इनामेल चाहिँ प्रोटिन कोड गरे जस्तो हामीले चाहिँ लिभर भनौँ न लिभर कन्जिनाइटल एमोरोसिसमा देख्यो नि अब त्यो त्यस्तो टाइपको यदि हामीले मेबी न्युक्लिक एसिडहरू इनकोडिङ गऱ्यौँ भने त्यो एभीमा मेबी त तिम्रो आउन सक्ने चान्सेस छ Isa, if you have, you can add up है त
त्यही हो मेरो नि
अनि एकचोटि त्यो डिएनएले प्रोटिन लास्ट एक्सप्रेसन त प्रोटिनको फर्ममा हुन्छ नि हैन इनामेल चाहिँ मिनरल होइन र
प्लस प्रोटिन
मिनरल प्लस प्रोटिन अनि अब मिनरल कहाँबाट आउँछ जिन थेरापी गर्यो प्रोटिन एक्सप्रेस भयो अरे हैन इनामेलको लागि अनि प्लस हामीलाई मिनरल पनि त चाहियो
सो मिनरलको लागि चाहिँ मेबी आउटसोर्सबाट तपाईले जब के अरे मेबी इन्जेक्ट गर्न सक्नुहुन्छ अर मेबी त्यो त्यस्तो टाइप अफ डाइटहरू खानुहुन्छ डाइट रेस्ट्रिक्सनमा बस्नुहुन्छ भने डेफिनेटली आई गेस कि प्रोटिन त इन्कोड हुन्छ अनि त्यसपछि चाहिँ मिनरल्स एक्स्ट्रा साइडबाट या त त्यस्तै टाइपको हामीले प्रोटोकलबाट यदि मेन्टेन गऱ्यौँ भने डेफिनेटली हुन सक्ने चान्सेस छ बिकज
मलाई चाहिँ के लाग्छ भन्दाखेरि मैले अघि भने नि एबीमा लाइक 3टा प्याकेजिङ प्लास्मिड हुन्छ अनि 3टा इन्कर्पोरेट गरेर मिल्छ भनेर सो मेबी तिम्रो अब एउटामा प्रोटिन लाइक जुन चाहिँ तिम्रो इन्कोड गर्नुपर्ने हो प्रोटिन त्यसको जिन हाल्यो एउटा प्लास्मिडमा अनि अर्कोमा चाहिँ चाहिने मिनरल्स हालेर पनि गर्न मिल्छ नि त त्यो पनि एउटा हुनसक्छ भन्ने फेरि
यस्तो भएको छ कि नै
तिमीहरूको इनामेल भनेको फर्स्टमा तिमीहरूको लिभिङ टिस्यू हो कि डेड टिस्यू हो त्यो थाहा हुनुपर्ने वाट यु थिङ्क इनामेल भनेको के हो लिभिङ हो कि डेड टिस्यू हो भनेको डेड नभन्नु लेट्स नट से डेड लिभिङ हो कि नन लिभिङ हो कस्तो हो यो इनामेल
नन लिभिङ हो
क्याल्सिफाइड टिस्यू
ओके नन लिभिङ हो होइन सो आर्टिफिसियल्ली इनामेल मात्रै ग्रो गर्न इट्स डिफिकल्ट है आर्टिफिसियल्ली इनामेल मात्रै ग्रो गर्न डिफिकल्ट छ बट रिसेन्टली पनि नभनौँ एकचोटि 2009मा तिमीहरूको चाहिँ एउटा युरिगन स्टेट युनिभर्सिटीले चाहिँ उनीहरूले त्यहाँ अब तुथको इनामेल कन्ट्रोल गर्ने जिन चाहिँ फाइन्ड आउट गर्यो भनेर एकचोटि चाहिँ तिमीहरूले अहिले पनि हेर्न सक्छौ यो साइन्स न्युजमा चाहिँ तिमीले यो यसो खोज्यो भने यु क्यान फाइन्ड इट है तिमीहरूले चाहिँ नि इनामेलसँग रिलेटेड वी हेभ जिन्स द्याट कन्ट्रोल द प्रोडक्सन अफ टुथ इनामेल भनेर चाहिँ त्यहाँ तिनीहरू चाहिँ एउटा पेपर पब्लिस गरेका छन् कि यो 2009 2010तिरको कुरा हो यो है त्यहाँ भन्दाखेरि त्यो मैले फेरि त्यसको डिटेलमा त म गएको छैन बट नर्मल्ली अब आफै चाहिँ नि यो नन लिभिङ टिस्यू भएको हुनाले चाहिँ नि ग्रो गर्न अलि अप्ठ्यारै भयो यु मेक इट केमिकल्ली होइन तर चाहिँ नि पछि चाहिँ साइन्टिस्टहरूले दे ह्याड क्लेम द्याट जिन्स दे फाउन्ड द जिन्स रेस्पोन्सिबल फर प्रोडक्सन अफ इनामेल भनेर उसले बनाएको थियो है ल यो भयो यो बाइकको अरू केही छ के अरे सुस्मिताले गरेर हेर न यो है तिमीले गरेर हेर्न सक्छौ है
हुन्छ
अनि ल ओके अब हामी के अरे सुस्मिताको सुरु गरौँ न त ल प्रेजेन्टेसन है
This is the part 2 of previously done presentation on COVID-19. We have previously dealt with the structure of coronavirus COVID-19 and how it infects our cell. And the main thing, the takeaway from that slide was the spike protein. Now we will be dealing about vaccines for COVID-19. Wait, it's not working.
के अड्कियो के भयो अड्किरहेको
It was muted, I guess. So why we need the artificially or artificial induced immunity? In the first place, we have natural immune system, which helps us to fight against the disease, but still we need vaccine assistance from the vaccines. It is because the natural immune system is very slow and the rate of replication of virus or any other pathogen is very huge, very high. So we cannot eliminate them effectively. So we need the assistance from vaccines. So for this COVID-19, we have set 2 goals. First one is based on safety. We are thinking of producing that kind of vaccine which is safe, means it doesn't produce any serious side effects. We can't, in this pandemic, we can't just say that we need vaccine with no side effects, but we can expect that it has we can expect that the vaccine having less side effects, like swelling on in the muscle of in the site of the injection, or some fevers. We can expect that for this emergency use. So the second is efficacy. The efficacy are of three kinds: to prevent disease, to prevent infection, and to prevent the severe disease or severe complications. For this COVID-19, we are concerning all about the efficacy that targets to prevent the disease. So what does the efficacy means? We are bombarded with the word efficacy, but previously in previous vaccinations or vaccines, we were talking about the effectiveness. But in this COVID-19, we are generally talking about efficacy. So it means that the efficacy is related to that result which is gathered from controlled trials. But effectiveness is that word which used to describe the monitoring of participants in real world. But efficacy is for that term when we want to emphasize that it is done in very controlled trials. Like we are taking the participant which are of certain age group, which are healthy and who have no serious complications. So efficacy generally deals with relative risk reduction in vaccinated versus unvaccinated arms in controlled trials. The formula to calculate efficacy is risk in unvaccinated arm minus risk in vaccinated arm upon risk in unvaccinated arm. This formula is shortcut. I mean, it is derived for short-term use, but the real formula is efficacy equals to 1 minus relative risk. We know the Pfizer has 95% efficacy, so we need to calculate it. And I want Devaka to assist me in doing the calculation. Okay, we have the formula efficacy equals to 1 minus relative risk. That is the real formula, okay? So Devaka, are you ready? Just grab the calculator and come.
Okay.
Okay, for Pfizer, the first dose was given to 44,000 people, and second dose was given to 41,000 people. But we are considering that the sample number is 44,000. Yannick to 44,000. Write it down in your paper. The sample size is what? 44,000.
Can you give me formula?
I will tell you. 1 minus relative risk. The sample number is 44,000. And after the second shot is given, the participants were called to check for COVID symptoms. So with vaccinated arm, we found out that 8 participants were COVID positive. So with placebo, it was double blinded, means the giver and taker of the vaccine were unknown that the injection contained vaccine or not. They were unknown about that. Placebo is just the replicate. It is done so that the vaccination trial remains unbiased. In Pulsiper, we just take the saline water. For Pfizer, we have taken the saline water. It doesn't contain the actual component of the vaccine, like mRNA for Pfizer. So in Pulsiper means we have known that in Pulsiper, it is saline water, so it has no vaccine component, means it is unvaccinated one. So COVID positive is 162. Write it down. With Pulsiper, it is 162. So now take the percentage. Now take the percentage of how many people get infected with vaccination. It's very simple. COVID positive of vaccination 8 divided by total participant 22,000 into 100. We got the how many percentage of people got infected with vaccination. Did you get it? So we get for vaccinated arm 0.036% risk. Devaka?
Yes, I'm doing.
एक्सक्युज मैले तिमीलाई यसो इन्टरप्ट गरेँ है मैले सुस्मितालाई देवकालाई इन्टरप्ट गरेँ तिमीहरूको चाहिँ यो विथ भ्याक्सिन सबैले सुन है विथ भ्याक्सिन टोटल पार्टिसिपेन्ट 22,000 छ होइन 22,000मा तिमीले भ्याक्सिन फर्स्ट ज्याब दिँदाखेरि फर्स्ट सर्ट दिँदाखेरि तिमीले के भन्यौ फर्स्ट ज्याब दिइसकेपछि तिमीहरूको 8जनालाई पोजिटिभ देखियो भने तिमीहरूले होइन अब यो 8जनालाई पोजिटिभ देखेको भनेको तिमीहरूको चाहिँ नि यु मस्ट मेन्सन है तिमीहरूले के भन्नुपर्छ भन्दाखेरि अब बाहिर अब फर्स्ट ज्याब दियो बाहिर गयो कतै कन्टामिनेट गरेर अरू ठाउँबाट कतै कन्टामिनेटेड भएर आएको हो कि अब तिनीहरूसँग चाहिँ अथवा चाहिँ तिनीहरू चाहिँ भाइरससँग चाहिँ नि एक्सपोज गराइएको थियो तिनीहरूलाई
थिएन
भन्ने उनीहरूले चाहिँ आफै गए ज्यादै गए उनीहरूले चाहिँ आफ्नो इन्भाइरोमेन्टमा दे गट इट फ्रम फर्स्ट ज्याब लाए तै पनि पोजिटिभ देखियो भने जस्तो भयो होइन होइन ल ल ल ठिक छ त्यसो भए You can continue है मलाई I got my answer.
So the risk for in Pulsiper group is found to be 0.74%, and risk in vaccinated group was found to be 0.06%. 0.036%. We can calculate the relative risk by dividing the risk in vaccinated group divided by risk in unvaccinated group. So it comes around 4.9%. So PKC equals to 1 minus 4.9 or 100% minus 4.9%. It showed that it is 90%, 95% efficient. So what does the 95% what does it mean to have the 95% efficacy for Pfizer? Anybody? Okay, I'll tell you. It means that if you are vaccinated, you are 95% less likely to have the COVID-19. And it doesn't mean that if you get vaccinated and there are 10 people, 100 people in that program, then 5 people will have the COVID. It doesn't mean that. It doesn't indicate the number. It is a very much individual thing. So we have to say 90% efficacy means 95% efficacy means we are less likely to contract COVID. So in this first figure, we have vaccine. We have vaccine that prevents disease but not infection, and this is the figure for that vaccine that prevents disease and infection. For COVID, it is the vaccine that prevents disease but not infection. It is not this one. So with the administration of COVID-19 vaccine, we will be able to decrease the height of severe symptoms, as we can see in the figure. So are effectiveness and efficacy same or different? I have already discussed it in previous slides. So there are examples of the efficacy rates of COVID-19 vaccine. For Pfizer, it is 95% efficient. For Moderna, efficacy for this vaccine is 94%, and for Johnson & Johnson, it is 66%. So my next question is, can we compare the efficacy rate of COVID-19 vaccines? I mean, are we really going to say that Pfizer is more superior than Johnson & Johnson in terms of efficacy? Anyone?
होइन होला मेबी किनभने एफिकेसी तिमीले भनेको जस्तो लाइक कन्ट्रोल्ड इन्भाइरोमेन्टमा भने होइन
Yeah.
त्यस्तो कन्ट्रोल्ड इन्भाइरोमेन्टमा भनेपछि त एफेक्टिभनेस कसको कति हुन्छ भनेर थाहा हुँदैन.
Yeah, the number for Pfizer group, the sample number also vary. Means for Pfizer it is 44,000. For Johnson & Johnson it is 60,000 people were enrolled for vaccine dose. And plus the Pfizer took the efficacy for after 7 days after second dose. Means it only checked for symptoms, whereas the AstraZeneca checked for PCR after 15 days of second dose. Plus, Moderna and Pfizer or BioNTech vaccines were conducted between August to November. So in this zone or in this time, the rate of COVID infection was very slow, and then there was no new variant, whereas Johnson & Johnson was administered during December to February, where the rates were rising up. Plus, it was done in South Africa and Brazil, where the new variant like B13551 alpha variant and P.2 variant were identified in that case. For Pfizer and Moderna, they were not done in those kind of variants. They were done in original virus. It's so that even if Johnson & Johnson is 64%, has efficacy rate of 64%, it is still effective because we have done this in new variant. And among 100% participants, 67 participants were found to have the new variant. But the vaccination was prepared for original virus. So it can give the insight that the vaccination or vaccine like this can work for variant as well, if the mutation doesn't occur in that way that we cannot recognize them. Variant of concerns are alpha B117, beta B1351, gamma P1, delta, it's a scientific name, B1617.2. So the typical timeline for vaccine preparation takes around 10 to 15 years. But for this COVID-19, we just took 1 to 2 years. We have done the preclinical trial in mouse. Mouse model. Mouse doesn't have as similar as same E2 receptor as ours. So we have to we have to construct the mouse transgenic so that we can get the similar symptoms or symptoms of COVID-19. So we have to prepare the transgenic version of mouse. In preclinical trial, we have injected the vaccine to the mouse, transgenic mouse, and we have done the phase 1 checking. We have done the phase 1 to check for safety. For this, 10 to 20 healthy volunteers were enrolled. And in phase 2, the phase 2 is done to check for efficacy. And around 100 to 200 patients were enrolled in phase 2. In phase 3, more than 1,000 people can be enrolled, and it is done to confirm the finding in large patient population to find out rare cases of side effects of that vaccine. And phase 4 is for real patient, real world scenario. As of Tuesday, June 9, 21, we have found that the number of vaccine in clinical development were about 105. And that of in preclinical development, it was 184. So the platform used by those clinical phase vaccinations were protein subunit, viral vector non-replicating platform, DNA technology, inactivated virus, RNA, mRNA, viral vector replicating one, virus-like particle, viral vector plus antigen presenting cell, live and attenuated virus, virus non-replicating, viral vector non-replicating plus antigen presenting cell. This platform were used, and the number of candidate vaccine for that platform is 33, and so on. It took 10 to 15 years, but still we managed to produce or manufacture the vaccine within the period of 1 to 2 years. It is all due to the fact that we were studying SARS and MERS from the very beginning, from 10 years, I guess, and we were known to the spike protein or spike of coronavirus. For SARS-CoV, when SARS-CoV-2 hit our world, we were known that we can tweak the spike protein of MERS or SARS so that we can make the spike protein for SARS-CoV-2, which can be used as an antigen in vaccination. So how was the COVID-19 vaccine developed so far? The first is we have done the researches, we have used the researches or studies from previous vaccination. And second is we have conducted the vaccination program and the preclinical trial, manufacturing trial, or manufacturing process, building the resources were done overlappingly. And we have invested in our mouse money and working time. So some of the COVID-19 vaccine are Pfizer buying, is the company name. Pfizer has produced the vaccination, Moderna has produced the vaccination, which are in mRNA platform. And Oxford AstraZeneca has produced the viral vector vaccination. It is a non-replicating type. Sputnik V also comes under viral vector vaccination, and doses are 2 for Pfizer, Moderna, Oxford, AstraZeneca, Sputnik, and 1 for Johnson & Johnson. So Sinovac is inactivated virus. It works under inactivated virus platform. So the dose for it is 2. Sinopharm is also 2, and Covaxin is also 2. The protein-based COVID-19 vaccine is Novavax. The doses for it is also 2. So for Pfizer, it is seen that it takes a lot of resources to store it because it needs the temperature of around -80 to -60 degrees Celsius, and to store it for 6 months. And for Moderna, it is less than Pfizer, which is good. It is -25 to -15 degrees Celsius. It means that the cost for this will rise because we need the storage building for them. But for AstraZeneca, it works in the normal refrigerator. So how the AstraZeneca Oxford vaccine was made. We take the adenovirus of chimpanzee. They were not exposed to human beings, and they are not immunogenic to us. So we can use it. We take the DNA of adenovirus. We just cut this part. I mean, not cut this part, and this is the gene. The purple color is the gene of interest. It is the gene of interest for spike protein. So since it is a non-replicating viral vector vaccine, so we have cut the part of adenovirus that is responsible for its replication. So it goes into the nucleus. It is not in the cytoplasm, it goes into the nucleus. But the AstraZeneca says that it doesn't incorporate with our DNA, human DNA. So it just transcribed without being incorporated in our DNA, and it gives rise to the mRNA. And it gets translated into the cytoplasm and gives the spike protein, which is responsible for immunogenic, uh, which will elicit the immunological response. So AstraZeneca Oxford is 2-dose vaccination. Means first dose was given at day 0, and the second dose was given after 28 days. And it was found that in Brazil, 9,000 participants were enrolled, and we got the efficacy of 62%. But in UK, we have 3,000 participants, but with some errors. For day 0, the AstraZeneca has given half dose of vaccine for its first shot. The error happened here. And for the second dose, it has given the full shot. So we got this number, 90%. It's quite head scratching because with full shot we got only 62%, but with half dose in first shot we got 90%. And USA is still not published it. So I have said that in Pfizer the placebo was saline water, but in AstraZeneca, we didn't give them the saline water. We just give them the meningococcal vaccine to replicate the pain, or to replicate the inflammation that the vaccine generally produces, because saline doesn't produce any inflammation if in the injected site. So viral vector vaccine, it is AstraZeneca one. So I have discussed it earlier in the previous slide, and mRNA based vaccine is of Pfizer or Moderna. So what it does, how they produce it is that they got the sequence in 10th of January. They got the sequence from Republic of China, and from that they can synthesize the mRNA sequence for spike protein, and they have placed that mRNA into the lipid layer, and they have injected it to the person. In the viral vector vaccine, it goes to the nucleus, but in mRNA-based vaccinations or vaccines, they go to the cytoplasm, and in the cytoplasm, they just get translated into the spike proteins, and the cell express the spike protein in the membranes, where the T cells are where the T cells binds to it, and the immunological response can be generated by that. So what's interesting about the mRNA vaccine is that previously it was not licensed for human use, but after the COVID-19, it was licensed for general public or human use. So the vaccine, how does the vaccine work? It is injected, and the APC antibody antigen presenting cell present the spike protein in case of mRNA or DNA vaccines, and it led to the activation of T cell. T cell differentiate into CD8 plus T cell, which is cytotoxic in nature, and CD4 T cell, which is helper in nature, and CD4 also activate B cell, and APC also activates the B cells. So B cells is differentiated into the plasma cell and memory B cell. So it gives its plasma cell secrete the antibodies which are of neutralizing nature. And they just, if we get the active virus, or if we get infected from the virus, the neutralizing antibodies which are produced from vaccinations are ready to attack them and clear all the viral infection. So the virus will not get chance to spread over our body. So it will decrease the severity of cases and complications. So immunological readouts. For generic purpose, I would like to give you some ideas like IgM and IgG antibodies are detectable within 1 to 2 weeks after the onset of symptoms, if the vaccination is done. Recent studies have shown that the magnitude of neutralizing antibody responses is positively correlated with disease severity. The more is the neutralizing antibody, the more is the disease severity. So we have AD5. It's a non-replicating type, but the problem with this vaccine, which use adenovirus 5, is that we are already immunogenic to it. We are immunogenic to AD5. So we are going to respond, our immune system is going to respond to AD5. But we don't want that, because we want the immunological response to our spike protein, not the AD5 one. So quality and durability is affected by the anti-vector immunity. So AD26, it is same. We are immunogenic to AD26. because we are already exposed to it, but chimpanzee adenovirus is very much it has given the satisfactory reason so far because we have not been exposed to them, and our immune system doesn't produce any response to chimpanzee adenovirus.
Hello, Susmita.
हजुर
तिम्रो नेक्स्ट क्लास कति बजेदेखि छ?
1 बजे
तिम्रो नेक्स्ट क्लास कति बजे?
1 बजे
3 बजे? त्यसो भए ओके, कन्टिन्यू गर।
1 बजे
1 बजे त्यसो भए मेरो क्लास चाहिँ मैले के गर्नु पर्यो? मैले चाहिँ क्लास सकाउने हो कि हाम्रो के गर्ने? व्हाट शैल वी डू?
6 बजे
तिम्रो
खाना पनि खाएको छैन।
ल भोलि गरौँला
5 मिनेट्समा सकाउँ, 5 मिनेट्समा सकिन्छ।
सिधै उडेसी सकिन्छ।
We stop here today. Should we stop it here? अड्की आयो नि तिमीहरूको। हेलो
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 प्रब्लम छ जस्तो छ नेटवर्क।
ओके सुस्मिता,
हजुर
त्यो कतिवटा साइड बाँकी छ तिम्रो अब?
6 10वटा 5वटा
कतिवटा?
हजुर 5 छ। भोलि गर्ने
छिटो गरौँ न त।
छिटो गर्ने
हुन्छ नि त, हुन्छ नि। क्लासमा गरौँला नि त, हुन्छ नि त।
हुन्छ
ल छिटो गरेर भ्याउँछु भने गर, छिटो गरेर भ्याउँछु भने गर।
छिटो भइहाल्छ।
ल सुरु गर।
So for in Nepal we have got the vaccination from China, from Sinopharm company, and from Russia called Sputnik V. So virus versus variant. We have original virus, and plus we have variant like alpha, beta, delta, gamma, kappa as well. So if we look at the graph, we can see that in February 7, it is at the peak and it drops down. It's for original SARS-CoV-2. The blue represents the standard SARS-CoV-2. And as time passes, it is decreasing. The infection is decreasing, but for variant, it is increasing. The condition is that we are adopting some protective barrier like quarantine, and plus we are not here in this slide, in this table, in this figure, we are not vaccinated, so it is rising. But what if we change our we replace our current behavior like quarantining, and if we just go around and don't practice the quarantine or protective barrier behavior like wearing the mask, it shows that after we withdraw that behavior, the normal or standard SARS-CoV-2 has had risen up, and slowly it was going down. But for variant, it is not going down. It was not going down for as of data of 18th April. Done.
सक्यो
सक-
thank you very much. It was one of the very good presentation. It was one of the very good presentations on COVID vaccines and all the virus. यो टाइम कन्स्ट्रेन्टले गर्दाखेरि तिमीहरूलाई मैले बिचमा सुस्मितालाई मैले बिचमा चाहिँ डिस्टर्ब गरेँ. आई अपोलोजाइज फर द्याट. अदरवाइज इन्फर्मेसनहरू सब राम्रो छ. अनि अघि मैले भने जस्तै ऋषभ र चाहिँ नि इसाला भने जस्तै इट्स भेरी गुड एनर्जी. अनि अलमोस्ट एकदम फाइन छ. तिम्रो चाहिँ अब ग्राफहरू टेबलहरूमा माथि तल लेबल गर्नेहरू कुरा नछुटाउने होइन. अनि टाइम म्यानेजमेन्टमा ध्यान दिनु पर्यो. टाइम म्यानेजमेन्टमा पनि ध्यान दिनु पर्यो होइन. बट तिम्रो चाहिँ एकदम राम्रो राम्रो कुराहरू इफिकेसीको कुराहरू होइन. अनि इफेक्टिभनेसको कुराहरू द्याट इज भेरी वेल सेड होइन. अनि चाहिँ यो प्रोफाइलहरू पनि अनि तिमीहरू चाहिँ यु ह्याड बेटर. अनि तिमीहरूको चाहिँ अर्को चाहिने भ्याक्सिनमा तिमीहरूले चाहिँ यु डिड यो बेस्ट है. यु डिड यो बेस्ट फ्रम यर साइड. अनि तिमीले एउटा कुरा चाहिँ मैले के देखेँ भने एडजुभ्यान्टको बारेमा कुरा देखिन् जस्तो लाग्यो मैले. हो सुस्मिता, एडजुभ्यान्ट राखेनौ? एडजुभ्यान्ट
छ यताभित्र यता
कता छ खोइ?
सबै स्किप गरेको स्किप गरेको थिएँ मैले. एडजुभेन्ट रिक्वायर्ड भन्नेवाला छ यसमा. प्रोटिन सब युनिट भ्याक्सिन.
ठिक छ एडजुभ्यान्ट्सहरू. किनकि तिम्रो चाहिँ हाम्रो भ्याक्सिन्सको हिस्ट्रीमा चाहिँ एडजुभ्यान्ट्स आर भेरी इम्पोर्टेन्ट होइन. दे हेभ देयर एकदम चाहिँ कहिले इन्फ्लेमेसन इन्क्रिज गर्ने, इम्युन रेस्पोन्सेसहरू बढाउने, दे हेभ देयर रोल्स. अनि एडजुभ्यान्ट्समा तिम्रो कस्तो इन्ट्रेस्टिङ स्टोरी छ भन्दाखेरि मान्छेले पहिला लिपिडहरू साबुनहरू ऱ्यान्डम कुनै सब्सटेन्सेसहरू भ्याक्सिन लगाए के अरे भ्याक्सिन सँगसँगै चाहिँ नि एडजुभ्यान्ट्स कुनै ऱ्यान्डम कुराहरू हाल्दाखेरि यत्तिकै ट्रायल गर्दाखेरि एकदम अर्ली डेजमा आई थिङ्क इट इज इन फ्रान्स हो कि कहाँ हो, त्यही होइन साबुन हो लिपिडहरू हाल्दाखेरि अरू कुनै चाहिँ हुन्छ नि इरेलेभेन्ट चाहिँ मटेरियल्सहरू चाहिँ एज अ एडजुभेन्ट युज गर्दाखेरि तिमीहरूको चाहिँ यो इम्युन रेस्पोन्स बढेको देखेर चाहिँ नि एडजुभेन्ट्स होइन युज भइरहेको हुन्छ. अहिले चाहिँ एकदमै पपुलर भइसक्यो. अहिले चाहिँ युज भइरहेको छ. तिमीहरूको चाहिँ मेरो विचारमा अहिले तिमीहरूको एमआरएनए भ्याक्सिनमा चाहिँ त्यो जुन लिपिड कोडको बाहिरको लिपिड छ द्याट हेज बिन युज एज एन एडजुभ्यान्ट भनेर भनेको छ है. ठिक छ, त्यो पनि ठिक छ, तिम्रो राखेको पनि ठिक छ. तिम्रो यो रेस्पोन्सेसहरू पनि एकदम राम्रो राखेको छौ. इट्स गुड. ठिक छ. अलिकति टाइम बढी चाहिने रहेछ नि त होइन. टाइम थाहा भएन हामीलाई होइन. तर छैन, इट इज अल गुड. इन्फर्मेसन इज भेरी हुन्छ नि ग्रेट है. तिम्रो माथिको ऋषभ र ईशाको पनि एकदम राम्रो थियो. तिमीहरूले चाहिँ अब त्यही हो जति दिदी राख्यो त्यति बढी सिक्ने त हो नि है. तिम्रो मैले मेरो ब्याप्टिस्टमा यदि अन्डर ग्रेजुएटको भन्दा तिमीले धेरै राम्रो स्लाइड बनाएको छौ ल है. यसरी सिक्दै जानु पर्यो, पढ्दै जानु पर्यो है. यसरी नै अगाडि बढ्दै सिक्दै पढ्दै गयो भने एकदम राम्रो हुन्छ. तिम्रो यु विल बी हुन्छ नि, यु विल बी वाट यु वान्ट टु बी होइन. तिमीले जेको लागि इफोर्ट के अरे पुटिङ गरिरहेको छु यु विल बी द्याट. ल त्यसो भए आज मैले इन गरेँ. तिमीहरूको बोथ प्रेजेन्टेसन्सहरू चाहिँ नि भाइबरमा ड्रप गरिदियो है त ग्रुपमा.
हुन्छ.
ओके ल, थ्याङ्क यू. हाम्रो ऋषभ, ईशा अनि चाहिँ नि हाम्रो सुस्मिता, थ्याङ्क यू अल. ल आजको प्रेजेन्टेसन भयो त. वेलकम ल है. अनि अरू अनि जाँदै गर्दाखेरि तिमीलाई म के भन्छु भन्दाखेरि आफूमा वर्कआउट गर्ने हो है. आफ्नो यस्तो हुन्छ कि तिम्रो आफ्नो चाहिँ मैले आफ्नो चाहिँ उस चिनेर मैले केमा वर्कआउट गर्नुपर्छ होइन. मेरो चाहिँ कहाँनिरबाट चाहिँ मैले मेरो आफ्नो कुरा सबै कुरा स्ट्रङ बनाउन सक्छु भन्दै आफ्नो चाहिँ त्यो विक पोइन्ट्सहरूलाई डाल्दै हुन्छ नि फिल गर्दै नलेजले फिल गर्दै होइन सबै कुराहरू चाहिँ बन्दै लास्टमा तिमीहरू यु विल बी वन्डरफुल अल अफ यु है. यो त अघिल्लोचोटि प्रेजेन्टेसन दिएको हाम्रो सुमित्रा हो कि कसले हो, द्याट इज अल्सो गुड है. ओभरअल तिम्रो अहिलेसम्म चाहिँ मैले तिम्रो प्रेजेन्टेसन हेरेँ मध्येमा अल गुड है, अल गुड. त्यसो भए आज मैले क्लास चाहिँ इन गरेँ है त. तिमीलाई ढिलो भइसक्यो होला. आज मैले तिमीहरूको स्क्रिनसट लिन्छु अनि क्लास चाहिँ इन गर्छु. ओके ल मैले क्लास इन गरेँ है तिमीलाई के भन्न छ कसैलाई के सोध्नु छ भन्न छ केही तिम्रो क्लास सुरु हुन थाल्यो अब अरू क्लास पनि सुरु हुन थाल्यो त्यही भएर है त मैले क्लास इन गरिदिएँ ल बाई एभ्रीबडी बाई एभ्रीवन हेभ अ गुड डे
Bye bye, sir.
Yes, sir.
बाई बाई बाई